Okay, good evening everyone and welcome to Futures webinar series. I'm Ivana Yevtovic, webinar coordinator. Tonight we will listen about neuroscience, brain development, plasticity and repairing nerve communication. Our guest, Professor Adema Ribic, is a molecular biologist turned neuroscientist studying brain development. She completed her undergraduate degree in biology at the University of Sarajevo and specialized in forensics genetics in Sarajevo, hoping to land a job with the International Committee on Missing Persons. Having no luck with that, she joined Max Planck graduate program in molecular biology in Germany instead. She obtained her PhD from the University of Göttingen and in 2011 commenced a postdoc position at Yale University School of Medicine. In 2013, she took on a new position in Tufts University. Since January of this year, she is with the University of Virginia, building up her own research theme at the Ribic Lab. Tonight, we will hear about her previous and current projects, and I want to say welcome, Professor Adema, and we are thankful and honored to have you.
Thank you so much for this lovely introduction. Like Ivana said, I'm happy to answer any questions in Bosnian or English. If anyone doesn't understand anything, please stop me, raise your hand or scream "Vichte," and I will just go over it very, very slowly. I will talk a lot about what I've done, my previous research and my current research, and there's a lot of really fancy techniques that I'm going to talk about, which is good for you because you're going to get to kind of learn about them, but I will go through everything very, very slowly. So, like I said, vičte ako nešto ne razumijete. Feel free to interrupt me. Today is actually Thanksgiving Day in United States of America. It's a holiday, so luckily I'm not working. And it's common to give thanks to people, past, present, anything. And I do want to give thanks to my family. Back, just like what Ivana said, I did my PhD in Germany. And when I got admitted to the program, molecular biology, they actually, my sister got a loan to send me to study in Germany. And yes, I'm still crying over that. Sorry about that. I'm very thankful for that because if there wasn't for that, I wouldn't be here today. Thank you.
We are really grateful for and really thankful, and thank you for sharing this with us. It's really beautiful. Thank you so much for sharing.
I was also on the waiting list, just for the students here. I was on the waiting list for that program, but in the end, no one asks you how you got in, as long as you got in. So keep applying, keep pushing, keep trying, you will get there. And I'm really happy to talk about all this today. Yes, I'm still very emotional. And even I chat a little bit about how it is abroad and how a lot of people don't know where Bosnia is. And it's very true. So even now when I'm introduced to audiences in United States, people usually just introduce me as a scientist from either Croatia or Serbia. No one really knows where Bosnia is. They usually just refer to it as stuff, like these people from these stuff here in the middle of the Balkans, and no one really knows where we are, but we know where we are. And we're still trying to put us on the map, and we're slowly, with the foundation that Adem started, we're really beginning to do that. And like Ivana said, I did my undergrad in Bosnia, and then I moved to Germany to do my PhD in molecular biology at the University of Göttingen. I got into International Max Planck Research School, and that kind of propelled me further and gave me an opportunity to go to the United States to do my postdoc. And even in Bosnia, I was always interested in how the brain develops, how do we behave, the way we behave, and why do we behave that way? Not just at a level of behavioral, like what are our motivations? I wanted to understand what are the genes that are enabling our brain to actually modify our behavior in response to environment. And this is a really age-old debate in the field of neuroscience. The debate of nature versus nurture, genes versus environment. So what is more important for our behavior? What is more important for our development? Is it the genetics? Is it everything that specifies us as an organism? Is that also specifying our behavior? Or is it our environment? Is it the way we are raised, where we live? What are we exposed to as children? What is more important into making us the mature people that we usually are, and what is more important in making us different as each individual is. And usually it's never one or the other, so it's always both. And my lab studies how both environment and experience, aka nature, how they interact with genes and define who we are, and how that happens at the level of neurons that build the brain. And I started this research really a long time ago, even during my PhD. And to do this research and to do any type of research, especially neuroscience, you always need a model animal or a model system. Using the model systems, we study these things, and then we kind of interpret the findings, and we try to generalize them to, for example, behavior. And for me to study how experience affects genes and how that affects brain development, I use the model system of vision. So I study how vision develops. All of us take vision for granted. We develop, so except for people who have poor vision like me, we usually develop with fairly poor vision, and then we forget about it because as babies, we just forget all those memories that we built across the first year or two. And then our vision starts slowly developing and sharpening. And you can see that here. A newborn wouldn't see this dog in front of it. It would just see shapes. Everything would be really, really dark. And then slowly, as a newborn develops its pathways in the brain and its eyes, it starts to slowly distinguish little edges and colors and little shapes behind the dog. At 3 months, it can kind of pick out the general shape. Like there is something in front of me. At 6 months, it's a little bit cloudy, but it can see that this something in front of it has ears, has maybe a nose, and there is something else behind this shape. At 1 year of age, children can generally see edges pretty clearly, and that also parallels the development of their motor skills. So at 2 years of age, they can see pretty okay, and it also parallels that they usually start walking by them, they start crawling, they start recognizing the world around them. But this doesn't happen just all at once. As you can see, it happens gradually. And during this entire time, you have to have normal vision. So if you block the visual input by any means, for example, if the child develops a cataract, or if it damages the eye, the vision will not develop properly. So if you deprive visual input by, for example, let's say the cataract, like I just mentioned, the vision through that eye will stay cloudy forever. The child will develop with very poor vision, and it will remain that way. And on the other hand, if you exercise the vision, if the child grows up in a different environment that is, for example, enriched for certain shapes or certain objects, they will tend to recognize those things better than children that haven't been brought up in that environment. Meaning that environment and everything that is directly around us really shapes exactly how our brains develop. And vision is our most used sense. So humans evolved in such a way that we use vision predominantly for everything. That's our main sensory input. And we make a lot of decision making based on what we actually see, both literally and metaphorically. And what I study is how that process happens. And during this development, development of children, and also during development of experimental animal models, there is a short window of time called the critical period. And this critical period is critical. It is very important for it to happen. And during that precise window of development in humans, it's 6 months to 2 years, it is essential that children have normal binocular visual input. So if they lack, if for example, you block one of their eyes, or if something happens to one eye, binocular vision will not develop, even if the eye recovers later, simply because the brain will not learn how to process the information from both of the eyes. It's called critical, like I mentioned, for a reason. It is absolutely essential that this happens at this point in time, and that at this particular window, we are exposed to the right environment. These critical periods do not happen in vision only. We also have critical period for hearing, critical period for language development. This is why a lot of us are struggling when we grow up to learn languages, but all our children usually don't struggle at all, and it's enough to just put them in front of the TV with a Spanish show and they will learn Spanish. Children learn really quickly by passive exposure, whereas we adults, we need to invest a lot of attention to learn at the same rate. And I will talk about that a little bit later as well, because that's what my lab studies do. So like I said, I'm studying this process here and how that occurs at the level of neurons that build the brain. So what happens in the brain? So what happens in the child's brain during this period? And that has been studied for a really long time. As you can see here, I'm citing papers from 1975. Yes, science existed even then. And people have studied how neurons, how the cells that build up your brain, how they change during that time. And neurons are very special. They're not just cells. They have a cell body, and then they have a bunch of processes everywhere. And they're using those processes, those little hands, to communicate with one another. And this is how information processing happens in the brain. This is how we see with the brain. All the neurons in our brain are processing that visual information. So as you can see here, this is a human visual cortex. This is the part of the brain that processes visual information. And all of these black shapes with these spider webs around them are neurons. And these spider webs are these processes that are used for communication. They have these contact points called synapses on them. And you can see that throughout the development, especially around this critical period, the density of these spider webs and the density of these proteins increases significantly. This is a lot. Compared to newborn, there's a lot of processes around here. And then it drops a little bit as we age, but this is known as the peak development of synapse, peak synaptic development. So this is the peak development of synaptic communication in the cortex. At the same time, different genes start being expressed, genes that actually support and promote development of that connectivity. And this is how that looks. So initially, all those genes that are promoting development of connections between neurons are expressed at really low levels at newborn at around 1 month. And then they slowly start going upwards. And around critical period, their expression is really high. They support all of that synapse development and remodeling that's happening that supports the development of function, and that slowly drops down. And this is also evident in vision, and this is also responsive. So genes and the structure of neurons is responding to the environment too. So if you block the visual input, like here, if you close one eye and leave the other eye open, if you study the structure of neurons in that visual cortex, you can see the neurons that are responding, that were supposed to respond to that eye that was closed, shrink. They're not as branched anymore. But neurons that are responding to the open eye really expand their processes. They dominate the other eye. And similar thing happens with gene expression too. If you close one eye and leave the other one open, and you study which genes, which synaptogenesis genes are expressed, you can see really large changes in expression of these genes here. So you can see this MD is monocular deprivation. That's the group that had one eye closed. And massive, like a large number of genes is changed between the normally reared and the deprived group. And what you see here is just a pool of many different mice, and the statistical significance when comparing those groups is denoted here. So, like I said, we're studying this in animal models. This, for example, was studied, it was a paper from cats, from what I remember. I work on mice all the time. And mice have a very similar development to us. So they actually are born with their eyes closed, and at about 2 weeks of age, they open their eyes. And after eye opening, their visual cortex goes through these same changes that we do. They don't see really well, and then the critical period comes along, and then they start seeing things, and then everything is fine, they're adult. Also, in mice, that all happens within a couple of weeks. So we don't have to wait for years to study the development. We have this window of time between 4 and 6 weeks where we can study how vision develops. At the same time, in mice, during this critical period, synapses, so those neurons branch in the same way and in the same amount like in people, which is, again, really great to study how that process is modulated by different genes. And another thing that is really important, and this is something that completely transformed neuroscience field when it was discovered, is that cortical inhibition, so inhibitory neurotransmission mediated by GABA, also increases during that time, and I will touch upon that later. So how do we study this in mice? mouse visual cortex is between, mouse critical period for vision development is between postnatal day 21 and postnatal day 35. And it's divided in a couple of different stages: early critical period, late critical period, middle critical period. Early critical period is considered those couple of days after postnatal day 21. And the peak of the critical period, when the brain is super plastic and the most plastic, most responsive to the visual environment, is between postnatal day 25 and postnatal day 28. When mice turn adults around 40 days of age, so a little bit over 1 month old, and they're considered fully adult when they're about 2 months old, around 60 days of age. So we study how this happens by manipulating different things that may or may not affect vision, and then we study what happens with mouse vision. To study that, we have mice that are watching different things on a computer screen in front of them. And while they're watching that, for example, these bars that are moving up and down in different angles, we are recording brain activity from their neurons. We record brain activity with so-called electrodes. So they're just tiny little wires or silicone bits that are able to detect those tiny little changes that are occurring in the brain where neurons communicate. And all of you that have had any type of electrical engineers, and also just everyone knows that brain communicates, it's just electricity. It's just changes in voltages, and this is kind of what we're detecting. And what you see right here, we place an electrode in mouse brain, and then we detect what's called local field potential. And this is simply brain activity changes in potential that we're detecting as neurons talk to each other. So my lab studies it this way, and we've done it for a long time. And we do this also in awake mice. So instead of anesthetizing mice and putting them to sleep, our mice are actually fully awake and they're immobilized. You will see that later in a little movie. Their head is immobilized over a ball, a styrofoam ball or a treadmill. So they can walk and they can run, they can roam, they can do essentially whatever they want as long as they're watching the screen. So they're fully awake, and that's how we mitigate any kind of weird effects that we may get from anesthetics. And that's how we also study behavior, because anesthetized mouse doesn't really behave. And we studied that during these different stages in the mouse cortex. And what we know is that when we measure changes in activity of neurons in normal mouse cortex responds strongly to the eye that is opposite to it. So if you record from the left visual cortex right here, it receives more input from the right eye. And when you stimulate the right eye and record the responses in the left visual cortex, you will get really large response right here. And if you stimulate the left eye, you will get a smaller response. And that's called contralateral dominance. And if you close the right eye, the left eye will start picking up, it will get stronger only during the critical period. And this is what we see here. So in a normal mouse, we have strong responses to the right eye, weak responses to the left eye. But if we close the right eye during the critical period, right here, these blue bars, we see a depression of responses to that dominant eye. And those responses between right and left eye kind of balance each other out, meaning that the brain is plastic and the open eye is starting to see more. It's starting to pick up the slack from the eye that was closed. And this occurs only during that short window during the critical period. So it doesn't occur before it or during the early critical period, and it doesn't occur in adult brain, mainly because the plasticity, levels of plasticity that are enabling these changes are not there just yet, not even in young mice. So like I said, we're interested in how this happens at the level of synapses, at the level of the connectivity between the neurons. And as I mentioned, GABA transmission, so cortical inhibition, sedatives, for example, they trigger the onset of that critical period. So if you have critical period that is occurring at exactly 25 to 28 days after mouse birth, if you administer GABA, if you administer diazepam to those mice, you can simply move that critical period immediately after you administer the drug. So it's a very, very potent way to trigger early brain maturation. And again, this is why we're studying it, because we wanted to see how the synaptic connectivity of these neurons that are mediating that maturation, how that develops and how that happens and what are the molecules that are mediated in that process. Within the brain, so the cortical inhibition is mediated by many, many different cells. And if you take a brain and you slice it and then you start detecting different cell types in it, like here, both in mice and in people, we have 6 layers of cells in the cortex, in the brain. And a lot of those cells are excitatory, meaning they release glutamate. And they're so-called pyramidal neurons. They're here in gray. And about 10 to 20% of those cells will be inhibitory interneurons. And a lot of those are so-called pervalbumin interneurons. They're called pervalbumin because they, when you stain them with antibodies against pervalbumin, you can detect them. And this is actually how we detect them. They're very sparse. They're not very dense in the cortex, but they're very, very strong. So they mediate really strong inhibition, and they can inhibit all activity when they're activated themselves. And on these neurons, they also have dendrites. They receive different synapses from these excitatory neurons around here. They receive synapses that are close from neurons that are all around them. And then they also receive synapses from the thalamus. Thalamus is a pit stop for visual information in the brain, in the middle of the brain. It's a nucleus, meaning it's a group of cells that is relaying visual information from the retina, from our eyes, to the cortex. And we know from previous work by other groups that visual experience, so normal visual experience, actually helps these neurons, these red neurons here, these inhibitory neurons, it helps them mature. And this is why inhibition is able to trigger that whole development of vision and maturation of the brain. What we're interested in is what happens at the level of synapses. So this is a synapse between 2 neurons. This little triangle here that is filled with these little circles is a presynaptic terminal that contains the vesicles. And like I just mentioned, they can be filled with many different neurotransmitters. In this case, what's depicted here are excitatory vesicles that are packed with glutamate. So when glutamate is released, they will bind to receptors here in the neuron that is receiving that information, and they will activate that neuron to send another signal. And different types of synapses can exist one next to another. And within these synapses, so they're not just, they're kind of built like this. And what's commonly depicted is empty space between the synapses, but it's not. That space, that little 20 nanometers, tiny little strip of empty space is called the synaptic cleft. And synaptic cleft is filled with proteins. It's filled with those gene products that are stabilizing synapses, and they're stabilizing these structures here. So this reconstruction, this is a model of how this space between the neurons looks like. And you can see here that it's really, really densely packed. It's like a lot of, it's like a little hamburger meat packed between 2 neurons. And it's filled with proteins. And these are the proteins that my lab is interested in. So we want to know what builds the synapse and how can we use that to repair brain function, especially after injury. Within this hamburger meat layer right here are many different proteins. And you can see them all listed here. Encadherins, ephrins, neurexins, SynCAMs, and all of these proteins are connecting one neuron to another, and they're enabling that communication between them. Also, a lot of these proteins, mutations in them have been linked to a number of mental disorders. For example, it is known that neurexins and neuroligin are linked to different forms of autism spectrum disorders, LRTM to schizophrenia, encadherin to many different things, ephrins also to schizophrenia, and SynCAMs to autism. My lab studies most of them, and we're primarily focused, and what we're going to talk about today, are proteins called SynCAM, synaptic cell adhesion molecules. Think of them as glue that literally glues synapses, points of contact between neurons together. And we study this in many different ways. So for mice, for visual system, when you close that eye in that experiment and you study what happens, SynCAMs act differently. So their gene expression changes. And you can see that here. So if we close one eye of mice and then we study gene expression and protein expression in their brain and we see what happens, we isolate the brain and then we extract the proteins. And when we run Western blot, and you can see that here, This is a control side of the brain, and this is the side of the brain that is responding to that closure. Only one protein actually changes expression out of all of these. Out of glutamate, GABA receptors, other SynCAMs, it's only SynCAM1 that actually increases its expression after eyelid closure, meaning that visual activity, or the lack of it, changes expression levels of SynCAM. So that's already kind of giving you a clue. This is how environment changes gene expression to affect function. So we wanted to know what that function is. So what happens to SynCAM? What does SynCAM do in the brain? SynCAM1 is expressed everywhere by all cells, and it's mainly situated at synapses. So it is expressed usually here in this hamburger meat layer. And we can use antibodies. They're tools that we use to detect different proteins in a tissue. And if we use antibody against SynCAM1, we can visualize it via different fluorescent markers that will kind of light up green under a microscope. And then we can see SynCAM here in green. And these black holes that you see are neuronal cell bodies. They're black because SynCAM1 is not expressed in the cell body. It's a synaptic protein, so it will be expressed at synapses everywhere around the neuron. And you can see one such neuron here stained with another antibody. This is a pyramidal neuron stained with μN. And SynCAM is also present at its dendrite, at one of its processes. And SynCAM1 is also present around and on inhibitory neuron, inhibitory provalbumin neuron, stained here in green. So SynCAM is present at both types of neurons, both excitatory and inhibitory, and it is situated, we know that from our previous work, at a certain type of synapse, at an excitatory synapse that releases glutamate. So, but where is it more important? So if we have this increase in expression after eyelid closure, where does this increase happen? Does it happen in excitatory neurons, or does it happen in inhibitory neurons? And when we repeat this same experiment and then we quantify the signal, this green signal of where is it changing, we can do that. And you can see that here during critical period, if you close one eye, and this is a control hemisphere, this is a hemisphere that responds to that eyelid closure. These are the pyramidal neurons in gray, and these are the provalbumin neurons in red, and the green is the SynCAM1 signal. And then we quantify the signal over each process that a neuron has. And we compare it between many, many mice and many, many images, and you can see that here. So these are two different groups of mice and two different groups of data, a control hemisphere and a hemisphere that is responding to the closure. And these little asterisks indicate that the change in expression was significant only when signal was near provalbumin interneurons. And that gave us a really good indication, okay, when we close one eye, when a change in environment is affecting genes, when it's affecting SynCAM1 in the visual cortex, that is restricted to inhibitory neurons. So what does that mean? What does SynCAM1 do in provalbumin neurons? We didn't know that, and it's really important. Like I mentioned, inhibition regulates plasticity and brain development, so we really wanted to know whether SynCAM1 is important for that process. So what happens when you remove it? And we can do that using knockout mice, so we can develop mice that simply lack the gene, and then we can quantify different types of synapses in the brains of those mice. And you can see that here. Every little red cell that you see here is a provalbumin neurons, and these little blue dots that you see around them are synapses. They're really, really tiny. They're around 200 nanometers, so 0.2 microns. They're very, very tiny. And then we can quantify them by simply counting the number of dots that are contacting each dendrite, each process of this provalbumin neuron. And in blue here, these are synapses that are coming from all of these gray neighboring neurons. So all of the neighborhood neurons in the brain that are synapsing, that are contacting and talking to these provalbumin neurons. And we quantify the synapses between normal mice and mice that lack SynCAM1 over different ages. And what we saw was that actually there was no difference. So there was absolutely no difference in synapse number. Everything looked perfectly normal, which made us wonder, okay, maybe it's not these synapses, these are just local synapses coming from the neighboring neurons. What if it's the other synapses? What if it's those synapses that are coming in from the thalamus, and we can label them again with another antibody, and you can see that here in green. So when we label those thalamic synapses, and they're a little bit bigger, so these terminals, these projections that are coming from the thalamus to the brain, they're very, very big, and they're actually really a lot easier to see under the microscope and to quantify. And this is where we began seeing differences. So the SynCAM1 knockout mice initially don't have that many differences in the number that these axons, the number of synapses that these axons form with these inhibitory neurons in the brain. But as the brain matures, so during the critical period, the number of synapses starts dropping. So it never really catches up with normal mice. And you can see that here. Early on after birth, at the beginning of the critical period, normal mice have kind of a no change in the number of these so-called thalamocortical synapses because they're coming from the thalamus and synapsing in the cortex. But SynCAM1 knockout mice start losing them. They're born with approximately the same number of synapses, but they're simply unable to generate more during development. So these parvalbumin inhibitory neurons start lacking these thalamic inputs that are conveying information from the eyes and sending it to the cortex. And that already gives you an indication, okay, if they lose synapses, if they lose that means to communicate with other neurons, that means that something is wrong with the function of the brain. So this was what we did next. So what exactly happens if you remove SynCAM1? It only in parvalbumin neurons, so only in these specific cells, and then you watch what happens. And we can do that using viruses. So viruses are luckily used as tools to study really cell-specific effects of something, of genes or activity on a certain function. And we developed, these tools are super developed. We can actually really zoom in on a single cell in the cortex or on a single cell type. And in this case, we removed SynCAM1 not from the entire mouse. So we removed it just from these parvalbumin neurons. So everywhere else it's expressed, everywhere else it's normal, because this effect could have also been due to something else that we failed to see. And you can see that here. So this turquoise signal here is the virus. It's being injected into the brain, and it spreads only locally within the visual cortex. And this is a mouse brain, by the way. Visual cortex is this area here. And this pink signal here is that electrode that we record activity with. So if we inject that virus to remove SynCAM1 only from those parvalbumin interneuron cells and only in the visual cortex, what happens? So we get the same thing, luckily. So we get less of those thalamocortical synapses coming into the parvalbumin interneurons. Again, you can see here, this is a parvalbumin interneuron, and in green are those synapses coming in from the thalamus, and this is just the virus signal. And in a control mouse that didn't have any SynCAM1 gene silencing, the number of synapses is normal and very comparable to just a regular non-injected mouse. But in the SynCAM1 knockdown mouse, so in a mouse that had SynCAM1 removed only from those inhibitory cells, the density of those synapses is changing. So the number of these green dots that are around the parvalbumin neurons decreases sharply, and even more than in the general SynCAM1 knockout mice. So the effect was even stronger. So what happens to the function? So how do these mice see? Do they see is with electrophysiology? So this is when we put the electrode in the brain and we record brain activity while mice watch something. And this is how we do it. So our lab uses not just one single wire. We use what's called a silicone probe that has multiple different sites that act as electrodes. So we can record throughout the entire brain and get information from all layers. And this is how that looks like. It's a tiny, tiny little thing. It's only 15 microns big, so it doesn't damage the brain at all when you put it inside. And we can focus only on layer 4, which is the layer where all of these synapses are actually synapsing with parvalbumin neurons and with the excitatory neurons. And then we get the response, the signal that is called local field potential. And then from the local field potential, we can analyze it and get activity of single neurons. So what you see here is single neuron firing action potential. It's a single neuron talking to other neurons about what is it doing. And every single time you see a dot, that's an action potential fired. And you can see here, every single time you flash light in front of mouse's eyes, neurons start firing. They start really being active and they start talking. And this is kind of how the brain works. We see many different things, we hear many different things, and then neurons that are specialized to hear and see only those things begin to talk to one another. And this response, this strong response, is called a primary response. And then they don't keep talking all the time. After the stimulus ends, so after you turn the light off or you turn the screen off, the neurons kind of slowly start going back to baseline. And this is what you see here. So there's no dots, there's no talking. And this particular return to baseline, so return to normal activity, that's something that's mediated by those probably neurons here. So when the light, when you see something that activates your brain, these synapses here from the thalamus will convey that, they will transmit that information to your brain, to your cortex. And in the cortex, they will activate both of these cell types. They will activate both inhibitory cells and excitatory cells. But they will activate these inhibitory cells more strongly. Those synapses are bigger, they're stronger. So when they're activated at the same time as the other cells in the cortex, the inhibition immediately shuts the signal down. And this is why you have such a clean shutdown of the signal. You have this initial response, it really peaks after the stimulus, and then whoop, it immediately turns off. And this is essential to not exhaust neurons. So you have to do it this way. Otherwise, you would simply exhaust them metabolically, and you would also, your brain would be hyperactive all the time, and you wouldn't be able to distinguish what's really happening. You wouldn't be able to see all that well because your neurons would constantly be working. And that's never good. So what happens if you lose these inputs? What happens if you lose these inputs that are coming from the thalamus and the parvalbumin cells? What happens if you lose that activation of inhibition in the cortex? In the normal mouse, so this is a wild type mouse, this is a young mouse, you can see a neuron firing here. Trial is simply a presentation of light, and each row is one neuron firing every single time when the light is presented. And you can see that here, it spontaneously fires an action potential, but each time it sees light, it fires a lot, and then goes back to nothing, and then slowly goes back to pre-stimulus baseline. So if you remove inhibition, they constantly fire. And this is an adult mouse. This is a control mouse with SynCAM1 expressed in parvalbumin neurons. And you can see again, neurons are firing every single time. They're talking all the time. They're always chit-chatting. You present them with the visual stimulus. They will start firing a lot because that's their job. And then they will slowly go back to baseline. But if you remove that inhibition, if you remove what's stopping that activity right here, you get constant firing all the time. And this is how that looks like. It doesn't look pretty. This is what we see in a lot of mental illnesses. We see aberrant neuronal activity that happens precisely like this. And when we can quantify that, we can quantify the number of times that the neuron is active and the spontaneous activity. So this pre-stimulus activity is severely increased if you remove SynCAM1, just that one gene from this one cell type in this one brain region. This is what you get. You get a mess. So this is how important they actually are. So this was, for example, we removed SynCAM1 all the way from early development. So we injected this virus at about 2 weeks of age when the mice opened their eyes, and then we recorded this activity from mice that were adult. So what can we use this? Can we do this actually in mice that are already old? So can we just acutely knock down, remove SynCAM1 and get the same effect? And this is what we did. So we injected these viruses to remove SynCAM1 from parvalbumin neurons when mice were actually much older. They were about 45 days. And then we also studied their activity. Another, which is, sorry about that, we can see that here. And what we wanted to see is what happens to plasticity. So if you close one eye, and remember you record brain activity, will this removal of inhibition restore the young brain? Will activity be restored to those young levels because the brain kind of prevented from being matured? And this is what we see here. So when we record activity of the brain, in normal mice, they have that really, really strong response to the opposing side eye and really weak to the same side eye. And in mice that had the SynCAM1 removed only from those cells from a brief period of time, even when they were adults, so the virus is injected at about 45 days of age, we can see that the plasticity, so the eye will actually drop its responses to visual stimulus, and you can see that here. So the brain in these mice is as plastic as in young mice, as in the mice that are 25 to 28 days old. So these mice are old, they're about 2.5 months old, and just by injecting this virus that is carrying a tool to delete this gene from only a single cell type and only in a single brain region, we were able to restore the plasticity that would let the brain act just like a young brain after eyelid closure. And this was super important because tools that do this are highly sought after because they show promise in regeneration. One thing that is really, really difficult to do in adult brain besides learning is regenerating. So if the brain gets damaged, it's really difficult to repair those pathways that got damaged because there's the environment that the neurons are in is very prohibitive. So it's not letting them regenerate because of molecules like SynCAM. So if you remove SynCAM, you let them reestablish those connections. And if you can remove it just really precisely for like 5 minutes, just to let that happen, that can be considered a potential therapy. And this is what we thought was happening during development and also in adulthood. This is the brain, and these are the pyramidal neurons in blue, parabolic neurons in red. And in a normal mouse, in a control mouse, parabolic neurons are receiving multiple synapses. You can see them here. And SynCAM1 in red is connecting, it's right at those synapses, connecting these different things to parabolic neurons, sorry about that, and connecting those thalamic inputs with the parabolic neurons. So during the development, you have that rise in inhibition because of development of synaptic inputs of these cells that SynCAM mediates, and plasticity drops after inhibition is mature. If you lack SynCAM1, you lose these synapses, and inhibition never really fully matures. It stays at a really low level. Remember, you lose all of these inputs here, and then your brain stays like this. It stays really hyperactive, which is how it looks like right before eye opening. And then it stays at that level, but it also stays plastic for a really long time. This is good and bad. I will talk about why it's both good and bad in a little bit, but SynCAM1 is essential for this process, for brain maturation, and it is absolutely, what is really interesting is that it's acting in a very synapse-specific manner. So it's acting on this just one single type of connection on one single cell type in one single spot of the brain. And these are the tools that we're looking for all the time for repair brain connectivity. So what does this mean for learning? Like I mentioned before, I kind of threw myself into neuroscience to understand how do we behave, why do we behave the way we do, how do we learn? So how do we adults learn? How can we just go out and learn a new language, learn a new skill? What genes mediate that process? And we know, for example, for SynCAM, it has always been confusing. Mice that lack SynCAM are really good at some things. They learn really quickly some things, but they're terrible at others. So for example, they're completely unafraid and they're unable to learn to be afraid to certain things. One thing that my lab uses to study learning is classical conditioning. And all of you that have any pets will recognize this old experiment from Pavlov, Pavlovian conditioning. And this is still used to train different animals, to train dogs, to train cats, to train mice in my lab. You essentially condition an animal, condition the dog in this case, to respond to completely unrelated and unnatural stimulus. And in this case, a hungry dog will start salivating, will start producing saliva when it sees food. But if you couple presentation of food with a metronome, or with a whistle, or with a bell, the dog will start associating those two things together. And then it will start salivating. It will start thinking, oh my God, I'm getting food. Exactly when you present it with that thing that you conditioned it to, with a metronome or with a whistle or with a bell. And this is how we train dogs. For example, in real life, when we have a dog that we want to teach how to sit or how to lie down or how to go outside, we give them a food reward every single time they perform a behavior that we think is correct. And then we give them that reward. And then after time, they will learn how to do that behavior when we just signal them with something else, with a hand, with a whistle, with a clicker. It's used all the time. Mice can also be trained they can also be conditioned. And in my lab, because we're studying vision, we're training mice to discriminate between two different images. And this is a very dull experiment, but it's also very interesting. I think my sister called me one time when I was doing this, and she laughed a lot because it does look very, very silly, but it is very important, as we will see in a second. So we essentially have a mouse that is awake, and the mouse is right here in front of a screen. I will play this movie for you. You can see it here. It is walking on that little treadmill, and it's seeing different images. And whenever it sees a certain image, in this case, bars of disorientation, it will get water through this little spout. So if you can kind of see right here, every single time it sees something moving, it will lick that spout. That's it. That's the reward. So the reward for the mouse in our experiments is water. These mice really, really like water. They want water because they're really thirsty. And you condition them to recognize only this one orientation, but not the other, because you're not giving them any reward when this orientation comes on screen. So it's just presentation of different bars. You present bars like this, you give them water, bars like this, they get no water. And mice pick this up really quickly. What we're measuring is the licks. So how many times mice lick this spout. And they learn pretty quickly, to be honest. So these are the licks. This is how we quantify them. Every single time a mouse licks that spout, that is recorded by our software. And then we count when they licked, how long they licked, everything. We count mouse licks. And before they're trained, so when you just start training them, so in their first session, you just put them on the ball and then you start you start giving them these presentation and the water reward, they lick all the time. They're really, really thirsty. Remember, they're water deprived, and they will lick throughout the entire session. When there is no reward, they will lick. When there is reward, they will lick. They will just lick all the time. And over time, so in about 7 to 10 days, they will kind of start clicking. They will realize, oh, wait a minute, I have this bar on the screen. This is when my water will come. And you can see here, they start licking faster before the actual reward. So they start to associate this orientation with water. So they already know that it's coming when they see it on screen, and they show what's called anticipatory licking. So they're really happy. They're like, oh my God, the water is coming, and they immediately start licking. And we can see that here. And they stop being so stupid to lick constantly, and then they simply usually stop licking altogether. This is an average mouse. They stop licking when the reward that when the on screen is an image that they do not get a reward for. So like here. So before the training, they're licking indiscriminately. After the training, they restrict their licking only to when the reward is presented, only to this type of image. And we can quantify that. We counted the number of licks over time, and you can see that here. So this is a normal mouse, an adult mouse, old mouse, actually 3 months old. And you can see that the number of licks is increasing for the rewarded orientation over time. Trials is simply counting each time a stimulus like this is presented. And then in red, this curve that kind of drops, it signifies the reduction in licking when the non-rewarded orientation right here is presented. So the mice are actually learning that the water is associated only with this type of an image, but not with the other one. Like I said, they do this in like 7 to 10 days. Some mice need a little bit more time. So you usually let them try to learn this until 2 weeks. If in 2,000 trials they haven't learned it, they're non-learners and they will likely never learn. The older you go, the more harder they learn, and younger mice, we actually still don't know how they learn. we wanted to compare so SynCAM1 knockouts, the mice that lack SynCAM1 that show increased plasticity. So can they actually learn quicker or worse? We simply didn't know. So we compared normal mice and SynCAM1 knockout mice in this task. And actually, this was surprising, and this is a result that we just recently got from a rotation student from the quantitative neurobiology behavior program. SynCAM1 knockout mice can learn this task in a half or a third of the time that normal mice do. So you can see this curve for the non-rewarded orientation sloping down a little bit at the 1400 trials. In a SynCAM1 knockout mice, that curve starts sloping down at 300 trials. So these learn in 7 to 10 days. These mice learn in 3 days. They're done. They see it a couple of times and they're like, I have this, I got this. And this is quantified here. So a session is essentially a day. We have only one session per day. And for a wild type mouse, it usually takes them like the medium, like 10 to 15 days, 1 week to 15 days. After that, they're not going to learn. 13 is the mean value for this. And for the knockout mice, it usually takes them about 4 sessions to learn this task. What we're interested in right now is how these changes happen. So how does SynCAM do this? Which cells is it acting on? And what happens precisely during learning that enables this to happen so quickly in SynCAM1 knockout mice? To do that, and this is illustrated here, so how that visual activity, how that visual learning shapes the connectivity between the neurons. In a normal non-trained mouse, we think that certain synapses are really responsive to one orientation versus another, and then that changes with learning, but we don't know. And we do think that SynCAMs are, for example, redistributing in synapses during the learning process. And we're studying this with 2-photon microscopy. It's a technique with which we can actually visualize single neurons in the brain. And each little, so this is a dendritic branch, this is an arm of a neuron, and each one of these little circles is a synapse. It's a point of communication between this neuron and some other neuron that you cannot see here. And we can quantify how these little circles behave during learning in awake mice and see what happens exactly during the learning process and how SynCAM1 contributes to this. Another thing that my lab is interested in are neuropsychiatric disorders. So we're studying how abnormal experience affects brain development. And one of our models for abnormal experience is preterm birth. So babies that are born prematurely, at least here in the US, about 10% of children are born prematurely. And from those 10%, 50% will show some sort of cognitive impairments by the time they're adults. Meaning that something happens. So the preterm birth, that those couple of weeks to 2 months that you're prematurely exposed to external environment is not good for you. And we're studying that in mice again, and we're able to induce birth in mice early, about 1 day, but keep in mind that 1 day in a mouse life is a really long time. They're adult in 60 days, so 1 day is kind of equivalent to a couple of weeks. And what we do is we inject a substance called mifepristone, which actually induces miscarriages in humans, and promotes preterm birth in mice. So instead of delivering them at 19.5 days of gestation, our mice deliver them at 18.5. So it's just 1 day. And you can see that here we study them at these points. It's 1 day shift to an earlier time point, and we did expect everything just to be shifted. So they're going to have a normal development of, for example, activity in the brain, but just a little bit shifted. Unfortunately, that's not what happened, and this is the interesting part. So when you do that, this is a normal brain before the critical period. This is a normal mouse. This is during the critical period. This is after the critical period, and this is the activity of neurons in the brain. And you can see how it kind of slowly really concentrates after the stimulus is fired. Remember, these are visual cortex neurons, so they're responding just to the visual stimuli. Preterm birth, they're already really focused on that before the critical period. So they're way ahead of time, which makes sense. They would be born prematurely. But when you analyze this further, what happens is that the signal to noise ratio, so the processing capability of their brains, instead of being better, actually deteriorates over time. So they start off really good. They start off, of course, advanced, but that processing capability deteriorates over time, and we're trying to figure out what happens and how this process affects learning. Like what Ivana mentioned initially, my lab is at the University of Virginia. I'm in the Department of Psychology. So we do study genes and behavior. I started in January, right before COVID. We actually moved at the end of December, like right as the news broke out that there's a new virus in China. Building the lab during this period has been difficult, but thankfully I have a really supportive environment, so they've helped a lot. And I was able to build my lab. So if anyone is interested, please go to my website. It's ribicneurolab.com. And then you can see what we do, how we do this. We share all of our analysis codes and most of our data online, and feel free to reach out if you are interested in any of these things. I'm happy to talk all the time. For the students that are in the audience, please consider graduate programs at the University of Virginia. UVA is the second best public school. So Berkeley is the only school that is beating us every year. So you can, that just tells you enough about the ranking. Psychology department, the department that I'm in, is really great. They have a PhD program that this is actually where I get my students from. And we have a couple of different scholarships. One of them is quantitative neurobiology and behavior, and the student whose data I just presented is from that program. And we have a recently started program called Expand that is essentially training students not just for careers to be professors, but also for scientists in government, in industry, et cetera, et cetera. All of our programs are really focused on trying to emphasize mentorship. Most of the students are co-mentored, and we do have a lot of foreigners. So please apply or ask about the programs. We do accept foreigners, like I mentioned, and this is a great opportunity for any students that's interested. Our application deadline yearly is December 1st. And thank you. I'm very happy to answer any questions, especially if anything needs clearing up. I know that this was a lot of information, but like I said, I'm happy to answer anything. And this is it.
Can you hear me?
Yes, I can.
We will ask everyone to write your questions, please. And
or ask them. I actually don't know if people have access to.
Yeah, actually, yes, I can give them. If anyone wants to talk, I can give you, permission to talk. So, before anyone writes, I just want to thank you for your time and for this presentation. It was amazing, and you are amazing. And thank you for this information at the end, for opportunities for our students, because that is what's the. I hope that some of them will use it. We have really great people and really smart people.
We do. Like I mentioned, and like you said, my undergraduate degree was from Bosnia, so I went straight from Sarajevo to Germany. It was really hard initially. I didn't know anything. Everything was new. Every single essay, every single piece of equipment was completely unfamiliar, but that's easy to learn. That is nothing, and we're all really, you know how stubborn we Bosnians are. Because of that, it really lets us learn pretty quickly. And please go out there, educate yourselves. So definitely push it. So we don't have any data on bad, I'm going to say toxic effects of SynCAM. We studied a little bit what happens when you apply just the molecule inside the brain and set on the neurons, and we know that that really also impairs the development of synapses. So neither having a lot of SynCAM nor having no SynCAM is great. Like I said, you can learn faster some things, but you don't learn fast some other things that are also crucial for survival. When you try to train mice to be afraid of something, opposite to what I'm doing, so in a sort of punishment behavior, mice that lack SynCAM never learn it. And that is actually detrimental for their survival. For the immunological portions of SynCAM action, there are multiple labs that are studying it. SynCAM1 is found also in cells in the immune system, and it's mediating an adhesion between cells in the immune system and, for example, different parasites. My lab doesn't study it. There are many others that do, and I'm happy to point you to that direction. And thank you. No, so Using models are a tricky thing in neuroscience for me particularly because SynCAM 1 is expressed only in vertebrates. So that already rules out fruit flies and nematodes like C. elegans. So we can study it only in mice, rats, and monkeys. But monkeys are really difficult to do research on because of all the ethical concerns. And mice and rats are easiest to work with in terms of genetic manipulation. And this is why we're doing them. Behavior is really difficult in both because they're not as trainable as, for example, higher mammals, but we do what we can. And yes, reward-based education is known, that's proven to work, definitely, especially now, be able to learn punishment-based learning mechanism. They just don't learn it, but they learn really, really fast the reward-based process, like the conditioning to water. That's always better. Yes. For the viruses that are used, that's a good question. We use specially engineered viruses that are not supposed to have any long-term or short-term toxic effects. So we're using adeno-associated viruses. They're engineered to kind of lose all and shed all those different things that make them infectious for the body. And AAVs are the ones that are used worldwide for development of gene therapies because they're non-toxic. There are many other viruses with higher toxicity that are used for very specific things like neuroanatomical tracing. My lab is also starting to use those, but those are not used in, they're not even considered to be used in things like, for example, treatment like AAVs are. Yes, so SynCAM 1 is thought, mutations in SynCAM 1 are linked to some forms of familial autism spectrum disorder. I know that there have been a number of reports for that, most situated at the synapse. So most of those proteins that are connecting neuromedication, they're mutated in both autism and schizophrenia and other types of mental illnesses. This is one of the reasons why we study what exactly do they do at the synapse, because all those processes are impaired in mental disorders, and we're trying to figure out, are there any other ways that are non-invasive, like behavioral therapy or pharmacologic substances that are already approved to kind of help them do their thing and mitigate those effects. Subjects for experimenting. Ethics is changing. So unfortunately, it is not changing in a good way. When I started my PhD was on monkeys. And I studied brain development in monkeys. I studied neuroimmunology. And it was a lot easier to do that 10 years ago. Now it's really difficult. A lot of scientists that are working with monkeys outside of China are sort of being exiled to go to China simply because of a lot of protests coming from the animal activist groups. Keep in mind that it's incredibly, while cell culture experiments and modeling are important for initial phases of studying how genes affect our behavior and how certain drugs affect our behavior, especially for drug testing, for testing new medication, you have to test it in animals because you always have to look out for those systemic effects that may happen. I personally would not try to use a medication that hasn't been tested on animals because I don't know what it would do to my other systems. And this is why animal research is so important. It is vital, and we really need people to support it. Otherwise, we will not get anywhere, especially with regards to neurodegenerative disorders like Alzheimer's and Parkinson's. So for Amar Big, which is asking for, okay, so for people, reward-based can actually be, for people and for animals that are able to distinguish different, for example, voices, the reward can also be just that. So when you think of the reward, it doesn't have to be food, it doesn't have to be water, it can be an achievement. Think about reward-based education. A child learns something, performs really, really well, and gets a grade. That's a reward-based system. A child really does something well, and the educator compliments the child on doing a really good job, that's a reward. And that's already a reward-based system. The punishment aspect of that is not great. So this is something that people always revert, in a lot of cases, people revert to, so they shouldn't. But at the same time, overt praise is also not great, because if you're overtly praising someone, you are facilitating those not so great behaviors, because we, when we're being conditioned to something, the timing and the precise focus on that really tiny little behavior that you're trying to condition for, that you're trying to reinforce, is super important. So reward-based learning or training has to be really, really well developed and designed to really work well. And I have questions in chat as well. Yes, so there are immune molecules within synapses. That was my PhD work. They are performing different functions to what they do in the immune system. So in the immune system, there are many different molecules that are signaling between, for example, T cells and B cells that are fighting against pathogens. But in the nervous systems, those molecules do different stuff. So instead of fighting off pathogens, they're actually signaling between the synapses. And this is where a lot of crosstalk occurs. And this is one theory of neurodegenerative diseases, like, for example, multiple sclerosis or even autism, for that matter, is that those immune connections begin signaling the wrong things. And this is why some of the really important connectivity gets destroyed. And this is how some mental illnesses arise. That's one theory about that. So this is what we're working on about the GABA question. That's a great question. So if you change the amount of GABA during development, that can have really detrimental effects for development of function. If you increase GABA early in development, it will simply open those critical periods early and terminate them early. And if those critical periods have to be timed well to the environment. So if you don't time that really well, that's not great. Now your functions do not develop properly. Also, some things that are not dependent on inhibition, but are, for example, dependent on proper development of some other things that are, will not get developed if you mess up, if you mess with inhibition. If you delay inhibition, on the other hand, so if you restrict it, you will not get development. With adult brains, that is one potential therapy that's currently being tested. We think that reducing the inhibition, reducing GABA signaling in adult brain can actually help the brain recuperate and can help learning. And this is one of the things that my lab does. A number of labs are doing this. It works. We know that, at least in mice and in rats. And we're still trying to figure out how exactly to do that so it doesn't interfere with other functions that GABA is needed for. And yes, so SynCAM1 mutations. So if SynCAM1, for example, let's say mutates in one of the domains that it uses to connect one neuron with another, if that's impaired, neurons will not properly communicate. And this is, for example, one reason why we had mental disorders. Synapses, so those contact points between your neurons, if they don't develop well, your brain doesn't function well. And it's not, your brain is simply sick and is unable to process all the information that's coming to it. And this is one of the hallmarks of those neurodevelopmental disorders like ASD and schizophrenia. Okay, so the mirror neurons. The mirror neurons are actually a characteristic of higher primates. Mice and rats don't have them. And I don't think a lot of people know that much about them. Simply, like I just mentioned, it's really, really hard to do these experiments in monkeys. Like I said, please keep all that in mind. We know a lot of things that we do right now thanks to animal research. So always keep in mind that it's really, really important. Mirror neurons do help. Like I said, they're found in higher primates, and they are thought to be important for some types of behaviors that are really specific for primates, like, for example, neuron behavior in humans. But unfortunately, we don't know that much about them yet, since they're found only in higher primates. I don't think that an equivalent of them has been found in mice. There are drugs for people that can increase brain plasticity, except that they have bad side effects. So we're always working on trying to find something that has less side effects. an antidepressant called fluoxetine. It's a selective serotonin reuptake inhibitor. It's used to treat depression. It has been shown to increase plasticity in people. So people have tried to use it as a treatment, for example, for blindness, for some of the cortical types of blindness that I talked about, like cataract and amblyopia. But fluoxetine will have other negative effects. So since it's antidepressant, it will affect behavior a lot. And this is something that people are trying to figure out. How do you get all the good things of manipulating inhibition without getting all the bad things, all the side effects that come with it? Oh, I think I've answered everything. If there are any questions about how to apply for programs, especially for students that are applying right now, I know the deadlines are coming up right now. Please feel free to ask as well. I'm happy to help and to provide advice on how to apply for them, how to pass admissions exam, how to interview for programs, et cetera, especially now while you have me.
Thank you so much. And thank you everyone for writing your questions. I hope you all, I'm sure you all got your answers, and this was really interesting. And thank you for sharing your knowledge and time with us. I'm sure everyone enjoyed.
One answer, Nero was asking, am I offering postdocs? Yes, but that depends on the funding stage. Right now, my lab is not hiring any postdocs, but definitely reach out later. Postdocs here are funded by external grants, meaning that my lab first has to get a grant from the government, and that's where I get the money to actually pay the salary of postdocs. So definitely reach out later. Elma, hvala. That's my schoolmate.
It's great that you have support from your family and from your friends, and that they are watching you here with us. That's really, really nice. It's
warms my heart too. I know.
Yeah, it really shows that beside being a great scientist, it shows that you are a great person too.
Thank you. And thank you, everyone. Thank you, everyone, for all of the great questions. Like I said, I'm happy to answer any at any point in time. There is one more to cover out. So for a psychology student interested in neuroscience, neuroscience is a large field of study. Psychology is kind of within neuroscience. So psychology studies the mind and the behavior, and neuroscience studies the brain and the behavior. So the only difference is that mind part, which is why we kind of like to talk about our department as the department that studies brain, mind, and behavior. For a psychology student who hasn't had exposure to, for example, animal work that has done work only with human subjects, like human testing and EEG studies, I do recommend simply reading up on neuroscience, finding a topic that interests you, and reading just textbooks on it to understand how the brain works and which techniques are used in it. The best way to get inside a PhD program is something what Ivana is doing right now. If you find an internship, a paid internship in a lab outside of, or even in Bosnia, like I did my internship during my diploma thesis in Sarajevo, and it worked. So as long as you can actually show that you have been inside a lab, that you have some sort of lab experience working in the lab on a daily basis, that is always a big ticket to end inside a master's or a PhD program. I do recommend PhD, not master's though. So PhD programs will simply equip you better for things than master's degrees. And yeah, I think I answered everything now. Hvala vam svima što ste došli. Yeah, mail definitely. Definitely, anyone feel free to reach out. Slobodno pošaljite email, odgovorit ću čim stignem. Jako mi je drago da sam ovo uradila. Vidim dosta Raje iz moje škole i dosta familije moje u publici, to mi je jako drago. I uvijek sam, whenever you need me to talk about neuroscience, I'm always happy to.
I nama je jako drago što ste s nama ovdje stvarno, i ljudi su stvarno, imate prelijepih poruka zahvalnosti i na informacijama na svemu, i većina ljudi je ostala do kraja, što znači da ono što pričate da im se svidjelo, da su sačekali da vidite odgovore na pitanje, možda što nije njima palo na pamet, sačekali su da
Dosadne vijesti bilo. Dosadne vijesti bilo, to je to.
Tako da, vijesti su prošle, sačekali smo lijepo da prođe dnevnik. Imate srce iz Beča.
Hvala.
Ovaj,
Ima nas dosta u Beču, a
pozdrav,
Beč ima
Evo. Da, zapravo